Another cool article on neural interfaces. The work I'm sharing with you today, it was done by Fernando Pesantes Torres, Susan Schaffstein, and their colleagues at the University of Albany. They developed a 3D flexible self-folding macroelectrode array, which is done to analyze electrical signals of organoids. This is important because traditional flat microelectrode arrays just can't keep up with 3D cell cultures. Usually, to get a good reading from an organoid, you have to slice or compress it, which only lets you record from one face. Here instead, they created a device that can wrap around the spherical structure to contact multiple points at once. Here is exactly how they built it. Using only four photolithography steps, they fabricated bilayers of polyimide and SU8 photoresist. The self-folding mechanism is driven by the stress mismatch between the two layers during curing. SUH shrinks more than polyimide, bending the device into a well shape. The degree of bending is controlled by UV exposure dose. 200 millijoules per square centimeter produces a fully rigid layer, while 80 millijoules per square centimeter leaves SU8 only partially cross-linked, generating the curvature. Gold electrodes are then coated with PEDOT, PSS, which is non-negotiable because it drops electrode impedance from above 1 megaohm down to below 10 kiloohms, minimizing noise and ensuring proper tissue contact. They first validated the device on 8-day-old cardiac neurogastruloids, ideal for initial testing because heart cells produce high amplitude, rhythmic signal easy to distinguish from the noise. They then moved to human cortical brain organoids up to 1,500 micrometers in diameter of 15 months old, successfully recording spontaneous action potentials and local field potentials. Organoids remain healthy on the device for at least a week. One imaging caveat is that while both materials are transparent, the PI SUA device produces green channel autofluorescence and absorbs UV, interfering with the immunofluorescence in the chip. Off-chip staining is recommended as a workaround. Ultimately, the team plans to integrate the pH, oxygen, and neurotransmitter sensors to create a complete real-time platform for studying disorders like Alzheimer's and Parkinson's. And I can't wait to see more progress in this line. I hope you like this study. See you tomorrow.